Very good day, dear listeners. This is Dr. Alok Tripathi, a pharmacologist having 14 years of teaching and research experience from India. Today we are going to see the topic pathogenesis of cell injury. Pathogenesis of cell injury, if you will see the different principles which are involved in it, the first and foremost principle is type, duration and severity of injurious agent. Means it depends upon which particular injurious agent is responsible to cause the cellular injury, for how much time cells or tissue are exposed to the injurious agent, and how much severely it causes an injury. That actually differentiate the pathogenesis of cellular injury. I wish to give an example of it, like deposition of beta amyloid causes the cellular injury. If you will talk about this, deposition will require a long duration of time, and as per as severity of it is considered, it is too severe to cause cellular injury. Specifically, if I had to tell, neurons, it causes an injury which result into neurodegenerative disorder commonly known as Alzheimer's disease. Another principle which involved in the cellular injury is type, status and adaptability of target cells. Means one principle will deal with the injurious agents and another principle will deal with the cell, target cell, which exactly result into cellular injury. So it depends upon which type of cell is involved in the injury, what's the status of that cell, and how much capability of it to adapt. As you know about the sensitivity of your individual cell has a different. So there are chances that one particular injurious agent to a particular cell will cause a severe injury, whereas the similar cell which will act to another target cell which is having the higher sensitivity, there are chances that it may causes the more injury to that cell. The third principle of pathogenesis of cell injury is the intracellular phenomena which is involved in the cellular injury. Like in detail we will see in this particular topic, the cellular injury occurs due to mitochondrial damage or due to cellular damage, or free radicals is involved in the pathogenesis of cellular injury. So these phenomena, intracellular phenomena, which is also very important to see which type of cellular injury and what's the severity of cellular injury will be there. Moreover, some morphological changes could also result over there due to cellular injury, like the ultra structural changes that occurs, or cell swelling that occurs, or there is a damage of nuclear membrane. These kind of morphological changes which are actually occurs, which has to be taken account while discussing with the pathogenesis of cell injury. If we will talk about in my last presentation, we had deal with the types of cell injuries like reversible cell injury and irreversible cell injury that we had discussed in detail. So pathogenesis also belongs to the particular which type of cell injury is there. Like A reversible cell injury is occur because of the decrease in the generation of ATP molecules. ATP, which is known as adenosine triphosphate, which is actually a source of energy, source of stimulation by which actually cells will do its function. And these ATP molecules are generated in mitochondria due to the breakdown of different nutrients with the help of oxidase enzyme. Now, due to some or other reason, if this ATP molecules will not be there, these cells are not getting sufficient amount of energy to do its metabolic function, to do its cellular function, and that result into generation of, or that result into reversible cell injury. We will talk about intracellular lactic acidosis. It is very well known fact that the acidosis, or I wish to tell you in a different manner, the acidic environment is not good for the aliveness of or normal functioning of cell. If due to some or other reason, if there is a lactic acidosis or deposition of materials will occur inside the cell which result into lactic acidosis or acidosis in the cell, which result into nuclear clamping. This nuclear clumping result into what? Result into dysfunctioning of your nucleus, which result into damage to the nuclear membrane, nucleus, and ultimately damage of the cell. Moreover, there are chances that the damage to the plasma membrane pump, like we do have on our cell membrane, the ATP dependent sodium potassium pumps, calcium pumps. What's the role of these pumps? If we will see about the cell, our cell membrane is made up of phospholipid. And if you will see about the cellular membrane, you will see that in the phospholipid membrane, the proteins are embedded over there. And these proteins are having several roles, like they act as an receptors, like they act as an ion channel, like they act as an transporter, like they act as an pump too. When you will see about the phenomena of action potential, you will observe that the action potential is generated over there due to depolarization of the cell, which is because of there is an entry of 3 sodium ion. If suppose this is what a cell, you will observe that there is an entry of sodium ions are there. More specifically, if I have to call, this is a 3 sodium ions entry will be there, and there is a which is known as influx of sodium ion. Then there are chances that the cell will go for the hyperpolarization, which is nothing but a exit of 2 potassium ion, which is responsible for the activation of voltage gated calcium ion channel, which result into entry of this calcium 1 ion. This 1 ion of calcium is responsible for the activation of cell. Now once the cell get activated, the cell need to be get activated again for its normal function, and for which the cell required to be get normalized again. And how it will be able to achieve that normalization again? With the help of ATP pump. These pumps are the proteins which are been available over the cell membrane. Whenever the action potential completes over there, the ATP molecule activate the enzyme, which further activate the sodium potassium pump, which result into exit of, which is earlier 3 sodium enters over there. So actually these pumps are having function to keep the ionic balance in the cell. So they do the exit of 3 sodium ion, which was earlier I told you there is an influx of 3 sodium ion for the action potential. Now there is an exit of 3 sodium ion will occur over there. There is an entry of 2 potassium ion, which I told you earlier. There is a efflux of 2 potassium ion is there, and there is a one exit of 1 calcium ion, and that is how the cell become normalized. Now you just keep one condition: if the cell is remain active. So for how much time it will remain active? Just I want to give an example about it to you. That suppose, which is the most important thing for our life to keep alive over the earth? No doubt, the first and foremost thing is oxygen. Which is the second most? If you will observe, second most important thing to be alive on this earth is what? Sleep. Anything which need to be work efficiently will need to be relaxed efficiently. You can talk about heart also. When heart is able to maintain the blood pressure, when it is able to constrict properly. Now suppose, just imagine if it is remain constricted, it is not dilated, does the blood will enter over there from atrium to ventricles? Not possible. So does heart is able to pump even if it is in a control contraction condition? No, not possible. And this is how sodium potassium plays a very important role to keep the cell alive and functional. Now if suppose this sodium potassium pump is not working, the cell will not be able to alive for a longer duration. Cell is not able to function properly. So next, pathogenesis for a reversible cell injury. No doubt these pathogenetics we will see in detail also. Just I want to tell you about what are the pathogenesis which are commonly relate with the reversible and irreversible cell injury. Now another one is reduced protein synthesis. Now whatever your cell is doing, you will keep in your mind that cell is functioning on the basis of protein. A single cell is able to synthesize 1,000 different types of protein, and each of the protein has a different role. Like you know about the enzymes. What are enzymes? Enzymes are nothing but the proteins which are present over in the cell and which are responsible for normal functioning. Probably you know about the receptors. What are those? Transporter. You know about insulin, how it works in the cell. Insulin will not going to do the metabolism actually. It result into synthesis of a new protein which is known as GLUT. This GLUT protein is a glucose transporter which will allow the entry of glucose inside the cell. Now if glucose will not enter into the cell, does energy will form? No. So protein synthesis plays a very important role. These proteins are responsible for number of cellular functioning. For the normal metabolic need, we need a protein synthesis should occur regularly. Now just imagine and this protein synthesis will be there over the ribosomes. These ribosomes are present in the cytoplasm. They are present over the ER, which is nothing but it is present over the ER. It is present over the mitochondria. If you will talk about ER, I am talking about the rough ER. There are two types of ER. ER means endoplasmic reticulum. A rough and smooth endoplasmic reticulum. Now just imagine, due to some or other reason, if these ribosomes are been disappeared, chances are there. As I told you in the earlier case, there is some ultra nuclear morphological changes could occur in the cellular injury. So ribosomes are damaged due to some or other reason, or they are not capable to produce a proper amount of protein or exact protein that result into reversible cell injury. Whereas the pathogenesis which are involved in the irreversible cell injury, The first and foremost is mitochondrial damage. We had known about it. I told you also about the mitochondrial damage, one pathway, that is apoptosis. You will find that mitochondrial damage can be enhanced due to the increase in the influx of calcium ion. If I have to give a best example about the increase in the influx of calcium ion result into cellular injury, the best example is excitotoxicity. We do have a number of neurotransmitters in our body. They are being called as a depressant neurotransmitter, which will depress where they will act. Example is GABA, gamma aminobutyric acid. There are mixed one which will act somewhere as an stimulant, which will act somewhere as an depressant, like acetylcholine, like adrenaline, like dopamine. Whereas one specific neurotransmitter which belongs to an amino acid group, it is what? Glutamate, which is known as excitatory neurotransmitter. Wherever it will act, it will do the excitation. And it is very well known fact that overexcitation result into increase in the influx of calcium ion, which result into neuronal damage. If you want to see it, the best example of it is your epilepsy. If you will see, the researchers had been found that if there is an overexcitement of neuronal toxicity will occur because of a overfiring of glutamate. And that's why you will observe epilepsy is the disease where the conversion will occur, but if it will persist for a longer duration of time consistently, that result into the damage of neurons, which result into cognitive impairment too. Activated phospholipase. I hope I will come soon with you with phospholipase in detail, but I wish to tell you your cell membrane is made up of phospholipid, and this phospholipid is there, and a particular enzyme, a set of enzyme is there. It's not only one enzyme, a set of enzyme is there, which is known as phospholipase. They are present in the cytoplasm. Normally they won't get overactivated, but just in a case example I wish to tell you about, if there is an injury, if there is an activation of phospholipid, it will do the breakdown of phospholipase. Phospholipase get overactivated, which will do the breakdown of phospholipid, which is actually the content which will make the cellular membrane. As we know, lipase, which will break down the phospholipid, it is an enzyme, so it will do the breakdown of cellular membrane, which result into cellular injury. Intracellular protease enzymes are also there. If they are get overactivated due to XYZ reason, there are a number of reasons that I have told you earlier in case of etiology in my previous presentations. The intracellular protease enzyme get overactivated, that result into breakdown of cytoskeleton, like in apoptosis. Cytoskeleton can damage over there because of the overactivation of protease enzyme, and this will activate because of oxidative stress and number of other things, which ultimately result into breakdown of cytoskeleton, which is present over the cell, and that result into the alteration in the structural of cell, which is responsible for the cellular injury further. Moreover, endonuclease, this is what another enzyme I taught you in case of apoptosis. You can see, you can refer my last presentation in which I discussed about the apoptosis. You will observe that endonuclease is an enzyme which will activate over there. This endonuclease enzyme is responsible for the breakdown of nuclear membrane, and that result into nucleus damage, and if there is a damage of nucleus is there, that result into cellular injury, and best example of it is apoptosis. Now we'll see in detail one by one different pathogenesis which will involve in the cellular injury in detail. The first and foremost pathogenesis of cellular injury is ATP depletion. What exactly occur in case of ATP depletion? Why there is an ATP depletion? The first and foremost video that I made on the cellular adaptation and cellular injury, in that I talked about the etiology of cellular injury. If you will see, ischemia is first and foremost reason I had given over there to have a cellular injury, or a hypoxia. What exactly happened? What do you mean by ischemia? Ischemia is a condition where the amount of oxygen required by a cell or tissue is not been fulfilled. Then the condition which will appear is known as ischemia. And if suppose these cells are not getting sufficient amount of blood, the ischemia condition, so the blood is actually transporting oxygen which is required for the metabolic need of the cell, so that develop into the hypoxia condition. Now what will happen in case of hypoxia? The mitochondria in the cell is responsible for the production of ATP molecules. And how does it will produce the ATP molecules? Because whatever the ribosomes present in the mitochondria in the villi over there secretes an enzyme, proteins, which is considered as an enzyme, which will activated in presence of oxygen. So if hypoxia condition is there, these enzymes which will not going to activate. Even research says that when hypoxia condition develop, these enzymes are not getting activated, cell will require energy. And how it will do that one? It will produce the oxidative stress or reactive oxygen species for a while to meet the metabolic need of the cell. But due to which what will happen? Due to which it will happen that oxidative phosphorylation is decreases over there. And that ultimately, if you will talk about the ATP molecules which are being synthesized from the cell through the mitochondria is also reduced. If the ATP molecule is been reduced, which is produced in the mitochondria, cell's metabolic need is not been fulfilled. Then what will happen if the cell's metabolic need is not been fulfilled? The sodium pump I told you in this section only, the pump are responsible to regain the normal physiological status of the cell again. If suppose, if you will see the heading of sodium potassium pump, you will see ATP dependent sodium potassium, or you can call it as an ATP ACE sodium potassium pump, which is responsible or which will be get activated only in presence of ATP molecules. And if suppose ATP molecules is not there, does sodium pump will activate? Does it will work? No, it will not work. Its functioning is get reduces over there. So what will happen? There will be more influx of calcium. If there is a more influx of calcium, there is more influx of sodium. Sodium, remember one thing, will always deposit more amount of water, then that result into cell swelling, edema. And if there is more influx of calcium, the more influx of calcium result into several changes in the cellular functioning. which causes deposition of it, which causes toxicity. If we will talk about, there is a more efflux of potassium ion will also be there, and overall these ionic disturbance in the ionic balance result into ER swelling, endoplasmic reticulum swelling, cellular swelling that you will observe, which ultimately a type of reversible cell injury. Moreover, this decrease in the ATP molecule synthesis result into increase in the anaerobic glycolysis. What is anaerobic glycolysis? The name itself suggests. Anaerobic means which will not require oxygen. Whereas glycolysis, the itself suggests glycogen lysis to produce more amount of glucose. So if anaerobic glycolysis will increase the glycogen quantity because it will break down. So glycogen quantity will reduce over there. Lactic acid synthesis will be more over there. These all both the things result into decrease in the pH of the cytoplasm. That will result into lactic acidosis. This lactic acidosis, I told you, for the aliveness of the cell and normal physiological functioning of the cell, it will require a neutral pH. Now, if pH is reduced, it becomes acidic. That result into clumping of proteins, chromatin. Nuclear clumping will be there. And because of that reason, your nucleus get damaged, proteins get damaged, ultimately result into cellular injury. Moreover, if suppose ATP molecules are not there, there is a detachment of ribosomes will occur over there. And all of us knows about that, what is the role of ribosomes in the cell. Ribosomes are responsible for the synthesis of protein. And whatever your cells are doing, it's because of protein synthesis. No, so protein synthesis will reduce, result into cellular injury. Another pathogenic pathway is the mitochondrial damage. We will observe that if mitochondria get damaged, that result into decrease in the production of ATP molecules. And we had seen in detail the different pathway which result to hit because of the reduction in the ATP molecules. And this reduction of the ATP molecule is always be in relation with the mitochondrial damage. If there is a mitochondrial damage, the ATP production will surely be reduced, as the mitochondria is a powerhouse of the cell where ATP molecules are being generated. Moreover, if you will see about, if mitochondria is damaged over there, that result into the activation of caspase pathway that I had discussed in my earlier apoptosis topic. This caspase pathway, if you will see, caspase is an enzyme which will activate over there due to damage or due to overactivation or due to alteration in the function of mitochondria. These caspase are of several types. 1 to 9 types are there in general. 3, 8 and 9 are the major types of caspase which are involved in the apoptosis function. They actually activate the endonuclease enzyme which will do the breakdown of nuclear membrane that result into nuclear damage. Plus, it will also activate the protease enzyme which are responsible for the breakage of cytoskeleton. So ultimately, if you will see about this caspase pathway which has been released from the mitochondria get activated, activate the several enzymes that result into damage of the cellular functioning and could result into apoptosis. Moreover, if mitochondria get damaged, automatically, it is very well known by the researchers, the production of reactive oxygen species has become higher if there is a mitochondrial damage. And this reactive oxygen species, the name itself suggests the highly charged molecules are there, known as reactive oxygen species. They are been overexcited molecules. These are been bombarded on the different cellular contents like lipids, like proteins, like DNA, and these species are responsible. We will see in detail ROS also, which is also one of the reason to have one of the pathogenesis of cellular injury, which result into lipid, protein and DNA damage, and ultimately result into decrease in the cellular functioning and cellular result into cellular injury. The another one is, if you will see about suppose the oxygen supply has been reduced, toxins or radiations are been there, you will observe that result into dysfunctioning of mitochondria, result into decrease in the ATP generation and increase in the production of reactive oxygen species, and these all result into cellular injury, very commonly known as necrosis. Whereas suppose the survival signaling of DNA or protein damage will be there, that result into activation or stimulation of pro-apoptotic proteins. I had discussed this pro-apoptotic protein like BCL2 family, BAC BCL2, which are being pro-apoptotic protein, which will get activated over there, and anti-apoptotic. See, we are having on our cell two types of protein, pro-apoptotic and anti-apoptotic. If the both are at functioning at a normal level, our cell function normals. But suppose if pro-apoptotic means the protein which are responsible for apoptosis, their quantity get increases, and the protein which are responsible for depression of apoptosis is reduced, that result into what? Mitochondrial leaking, that result into apoptosis. I had discussed apoptosis and necrosis in detail in my earlier presentation, types of cell injury. Another one is increase in the cytosolic calcium ion. If suppose entry of calcium ion get increases, this result into increase in the mitochondrial permeability. If the mitochondrial permeability is not selective one, could result into activation of different pathways. We will discuss in detail soon. Activation of multiple cellular enzymes. Now if you will see, you can see this figure, you are able to see if there is an extracellular calcium due to some or other reason, if its quantity get increases into the cell, it will enter into the mitochondria, it will be there with the endoplasmic reticulum, at both the places it will be released. So cytosolic calcium ion quantity get increases over there, result into number of enzymes get activated like phospholipase. I told you earlier in this section, phospholipase is an enzyme which will do the breakdown of phospholipid, and your cell membrane is made up of phospholipid. So ultimately that result into cell membrane damage. It will also activate protease enzyme which will be responsible for the breakdown of protein. When protein get active damage over there, that result into damage of cellular membrane protein, the damage of cytoskeleton protein, that ultimately result into disturbance of normal physiological structure of cell, that result into damage of cell membrane too. Another, this overactivation or entry of calcium result into activation of endonuclease enzyme. We had discussed it earlier that endonuclease enzyme is responsible for the breakdown of nuclear membrane, which result into apoptosis. It also result into activation of ATPase enzyme. This ATPase enzyme is responsible for the reduction of ATP molecules. If you will talk about mitochondrial permeability get increases, that also result into decrease in the ATP molecules, and we had discussed in detail about the ATP molecules in the last section. Reactive oxygen species, if you will talk about what is reactive oxygen species, oxygen derived free radicals are being called as reactive oxygen species. If you will see, free radicals are a special type of molecules that having unpaired electron in their outer orbit, and that's why they become very unstable. If you will see causes of free radical generation, if suppose there is a reduction and oxidation reaction occur into our cell, that result into free radical generation. If there is an ionization radiations are been there like X-rays, then also free radical generation is there. If there is an inflammation reaction is there, then also it result into generation of free radical. If there is metabolism of exogenous molecule, when your cells are digesting the exogenous molecules for the metabolism, then also there is a generation of free radicals, and for the metals also it result into generation of free radicals. Now, if you will see about the reactive oxygen species, if that will occur, how it will be there? Suppose radiation, reperfusion, toxins, all these result into what? All these result into production of reactive oxygen species. How? These radiation, toxin and reperfusion result into formation of superoxide anion. You are able to see over here in this picture. This is what superoxide anion. This superoxide anion is responsible for conversion to hydrogen peroxide. See, this is hydrogen peroxide normally being synthesized in our cell. Normally being synthesized in our cell. Normally being formed in our cells due to metabolism. We are having in our cells peroxisomes which contain catalase enzyme which is responsible for the digestion of hydrogen peroxide. If suppose that will not be there, this hydrogen peroxide is toxic for our body. That result into toxicity. It's a decomposition of hydrogen peroxide result in due to the glutathione peroxidase. I talked about you catalase enzyme. These will do the digestion of hydrogen peroxide into water molecules. And if suppose hydrogen peroxide is there, that result into hydroxyl radicals. These hydroxyl radicals will act over the phospholipid molecules. Lipid peroxidation they will do, and that result into damage of cell membrane, which we are able to identify with the help of MDA. Research, if you will see in research, we used to see about the oxidative stress by uh, counting the MDA level, malondialdehyde level. This production of reactive oxygen species also result into protein modification, which will do the breakdown or misfolding of the protein. You are able to see over here. Suppose I have to make a particular protein which is responsible for the metabolism, like oxidase enzyme. In place of that, it is proteins get misfolded and there is the formation of another protein which is not oxidase. Does it will do the metabolism? No. And that's why the ATP molecule generation will reduce. Moreover, this free radicals are the less number of electron in their outermost orbit. It is unpaired electron. Because of that, it is overactivated. That result into DNA damage too. This all result into what? Ultimately result into cellular injury. If you will talk about the membrane damage, you will see about your membrane is made up of phospholipid. Now, due to calcium, over calcium, due to bacterial toxin, due to lytic component, due to decreased oxygen production, what will happen? The membrane, plasma membrane get damage over there. Phospholipid get breakdown over there. That result into loss of cellular component that I told you in case of necrosis too. Moreover, if suppose like this membrane is there, cellular membrane is over there over the lysosomes also. These lysosomes autolysis I taught you in my earlier presentation. You can see over there. Autolysis is a cellular type of cellular injury where the layer get damaged and the hydrolytic enzyme are released over there. Now the same thing will happen, lysosomal membrane get damaged, digestive enzymes will result into digestion of its own cellular component, cellular proteins, that ultimately result into cellular injury due to membrane damage. Misfolding of protein. I told you that whatever your cell is doing, it's because of protein. Like I will tell you, my cells are producing energy. It's because of oxidase enzyme. Now this oxidase enzyme is nothing but a protein. My cells are able to take the glucose. How? Because of good protein. Individual cell is able to synthesize 50,000 different types or 10,000 different types of proteins. If suppose in place of formation of GLUT protein, which is responsible for the transportation of glucose inside the cell, it forms some other protein. So does glucose entry will be there? No. That ultimately result into decrease in the ATP molecules, that result into DNA damage, that result into cellular injury. Moreover, this misfolding of protein is there, or due to DNA damage, you will find that the activation of pro-apoptotic protein will be there, which result into cellular injury. And all these occurs because of mutation of gene. And that's when at a number of genetic disorder, even in research, you can see that number of genetic disorder which we will come across have a mutation of gene result into ultimately DNA damage, or misfolding of protein result into some genetic disorders. Thank you, student, for patience listening to this topic. I hope I had explained you in detail the different pathogenesis which are involved in the cellular injury. Thank you, students. Have a good day.